A laboratory monkey that cost 8,000 yuan has now been sold for as much as 230,000 yuan, prompting the media to suggest that efforts to replace animal experiments are well underway. Modern experimental science has introduced new methods such as organ-on-a-chips, OOC, and artificial organs, which will fundamentally change the animal experiment models that have been used for so long by humans. Within less than 3 years, the laboratory monkey that used to cost 30,000 yuan has soared to 230,000 yuan, becoming a strategic resource conserved by the biomedical industry. It costs roughly 8,000 yuan to breed one experimental monkey for 3 years. The reason behind the skyrocketing price of laboratory monkeys within a short period of just over 2 years is analyzed in the report. Since 2011, China has not imported any monkeys from abroad. However, the origin of the commonly used crab-eating macaque for experiments is in Southeast Asia, leading to an aging population in China in recent years, which in turn lowers fertility. At the same time, the inability to import monkeys from overseas has resulted in the genetic singularity of the domestic monkey population, leading to a decline in the entire population. Therefore, some professionals in the biomedical industry have stated that the high price of monkeys indicates the accumulation of various problems over many years. With experimentation being hindered by a shortage of monkeys, what will be the way out for preclinical research in the biomedical field? It's hard to find a monkey is also an opportunity for a change in research thinking. The concerns of academia over the laboratory monkey shortage caused by the exorbitant prices of such monkeys are understandable. This is because the issue has a direct impact on the outcomes of basic biomedical research, which is relevant to clinical treatment, drug and vaccine development, and all aspects of life science, all of which ultimately relate to human health and life. According to the results of animal experiments in biomedical research, non-human primates are the most effective for drug efficacy, metabolism, and toxicity, with the strongest cross-reaction across all animals. From both an effectiveness and cost perspective, the crab-eating macaque is the most widely used experimental monkey species in preclinical research. The exorbitant price of laboratory monkeys at present is first and foremost due to market supply-demand contradictions. On the one hand, since the outbreak of the COVID-19 pandemic, there has been an increased demand for laboratory monkeys. On the other hand, the crab-eating macaque is a nationally protected second-level animal, and its breeding requires an application for a breeding permit and a license for the sale of wild animals or their products, as prescribed by regulations. Also, when breeding, new monkey species need to be introduced from other regions or countries to conduct crossbreeding to maintain a high-quality monkey population. The other side of the supply-demand contradiction is that government regulatory authorities are gradually restricting the trade and importation of wild animals. In 2011, the State Forestry and Grassland Bureau issued the Notice on Related Matters concerning the quota for the management and utilization of laboratory monkeys for 2011-2012 years, which suspended the importation of crab-eating macaque sources from abroad. Breeding units requiring crab-eating macaque sources can solve this issue by importing them from domestic breeding units. At the end of January 2020, 3 national departments jointly issued the announcement on the prohibition of the trade in wild animals. Under such circumstances, some institutions have chosen to take risks and smuggle monkeys from Southeast Asia. However, this is not only illegal but also not a sustainable solution. Additionally, expanding the monkey breeding industry also faces challenges in terms of technology, management, industry establishment, and circulation, which cannot be solved in the short term. In fact, when the price of a product is abnormally high and inconvenient to use, it means that the industry needs to be updated and transformed, such as when incandescent bulbs are replaced by LED bulbs and copper cores are replaced by optical fibers. The dramatic rise in the price of laboratory monkeys is similar, but it involves a more fundamental problem, which is changing thinking and reforming scientific research methods. As early as 1959, the animal scientist Russell and the microbiologist Birch proposed the 3R principles of animal experiments, which are reduction, replacement, and refinement. Later, many researchers agreed and proposed the use of other methods to replace animal experiments in scientific research. This is not only due to the rise in popularity of animal rights and welfare ideology, which demands that humans treat animals with compassion, but also fits into the direction of modern scientific research development. Internationally, it has been proposed that modern and future scientific research will not necessarily rely entirely on animal experiments but can take other approaches. This also means that the transformation of modern and future scientific research will not only solve the problem of a shortage of experimental monkeys but will also result in a qualitative leap in research results, speed, and smooth integration with ecological civilization. Preparations are actively being made to replace animal experiments. Currently, in modern biological and medical research, there are two basic approaches to replace animal experiments. Firstly, the development of artificial intelligence, AI, and molecular biology, as well as the combination and integration of AI and biomedicine. Secondly, biomedical experiments can already be conducted using various types of stem cells to cultivate organoids and OOC, which have been proven to be similar in effectiveness to animal experiments, but several times faster and several-fold cheaper in cost and price. At the same time, the use of AI to replace animal experiments for research can also be fully applied and validated in the early stages of drug and biological product development. For example, in 2020, AlphaFold 2 achieved amazing results. In the 14th Critical Assessment of Structure Prediction, CASP-14, held from May to July 2020, most of the protein structures determined by this AI software were very accurate. Not only similar to the precision of those obtained by experimental methods but also far superior to other methods for resolving new protein structures. On the other hand, there are many ways to replace animal experiments with biological experiments, and they are becoming increasingly diverse. The first approach to replacing animal experimentation is the use of OOC, which can simulate various organs such as the brain, lungs, heart, kidneys, liver, prostate, blood vessels, arteries, skin, bones, and cartilage. Since all foods and drugs ingested by humans must be metabolized by the liver before being excreted, liver chips can be created by using liver cells from animals such as rats or monkeys and from humans. This not only allows for the testing of drug effectiveness and safety but also provides a low-cost model for studying liver dysfunction and disease mechanisms. In practice, liver chips have correctly identified 87% of drugs and have not made any incorrect marks for non-toxic drugs. Organs cultivated using stem cells can also replace animal experimentation. The smallest of these organoids are 1 mm in size, while the largest are several millimeters or even centimeters in size. Now, brain, lung, heart, kidney, liver, blood vessel, artery, skin, and bone organoids have all been developed and are being used in research. A new study shows that the effects of drug experiments on organoids are comparable to those of animal experimentation. Additionally, for cancer treatment, the prediction of clinical tumor effectiveness by targeting and chemotherapy drug sensitivity experiments based on organoids has an overall accuracy rate of 83.3%. It is predicted that, with the development of AI, OOC, and organoids, the experimental results will become even more accurate, ultimately achieving results comparable to animal experimentation. OOC and organoids are also more cost-effective. For example, a set of lung cancer OOC costs only 900 yuan in China, and a set of liver cancer organoids costs 6,500 yuan. Of course, AI, OOC, and organoids cannot completely replace animal experimentation for the time being because organisms have a comprehensive integrated system of physiological and metabolic pathways. And different animals have different physiological and metabolic pathways. Therefore, future biomedical research can combine AI, OOC, organoids, and animal experimentation. However, some countries are already preparing for the reduction and replacement of animal experimentation in terms of management and scientific certification. At the end of 2022, U.S. President Biden signed the Modernization Act 2.0 for the U.S. Food and Drug Administration. The law stipulates that new drugs can be approved by the U.S. Food and Drug Administration, FDA, without the need for animal experimentation. We need to have a change in our mindset and actions towards this. One feasible approach is to first use AI, OOC, and organoids in research experiments, followed by the use of low-level animals such as mice, rats, fish, amphibians, and reptiles, and finally, non-human primates such as crab-eating macaques in critical moments. As a result, the experimental dilemma of one monkey is hard to come by will be greatly improved. Experts predict that the annual demand for 150,000 to 200,000 monkeys in China may not actually arise, but instead, the use of non-human primates may be greatly reduced, leading to a significant decrease in the price of experimental monkeys. The most important thing is that the involvement of AI, OOC, and organoids in modern experimental science means that there will be a fundamental change in the way scientific research is conducted in the future. Animal experimentation, which has been used by humans for a long time, will no longer be the only method, and with the participation of multiple methods, scientific research will become more efficient, faster, and just as effective as animal experimentation. Of course, this will also be more favorable for protecting animals.